So welcome to our presentation for a workshop on machine learning and mechanistic modeling. I kind of lengthened it here slightly to mechanistic multiscale modeling because one of our emphases will be the importance particularly in health and disease, which is to say for applications to medical problems, for understanding things at multiple scales. And of course, machine learning recently has undergone something of a revolution with deep learning, which has now provided a variety of both tools and ideas for better understanding the brain. At the same time, the CNS meeting overall has always emphasized the ideals of mechanistic multiscale modeling with simulations being done from the level of biophysics, ion channels, small scale reaction diffusion, up through subcellular, up through full cells, and up to areas of the brain, whole brain, and spinal cord, and other parts of the central nervous system. So again, the emphasis on mechanism is because often we give drugs or called pharmaceuticals, or we may do stimulation, sometimes called electroceuticals, or we may do some surgery, such as a leucotomy, cutting white matter. And all this is at a relatively low level, but ends up influencing the whole brain. And how are we to scale these many levels and understand things? So here is page up. The one of the scale figures that I have used in the past, but very similar to scale figures being used for many years. I think the first clear example appeared in Churchill and Sinyavsky's book many years ago. We go from a spatial scale down to the molecular, which if one were able to do, which one is typically not able to do, the molecular dynamics would bring you down to the angstrom scale. And then as you go up, you even get a scale that goes beyond this, really beyond space, into cognition. One of the things that I think makes the brain particularly interesting as opposed to other organs of the body is that, in my opinion, there's really a lot of scale overlap, that things that happen at the dendritic scale and happen at the cellular scale cannot be readily encapsulated to simply include in the local area network or in the column or in the area of the brain, because there's so much interaction with, for example, in the neocortex, stimulation coming in, both high and low into apical dendrites, into basilar dendrites, and influencing the cell in very different ways. So this interplay between subcellular scale and network scale in that particular example. There are many such examples. So as I said before, healthcare really requires that we think about mechanism. We can't simply say, okay, we understand things at a very high level, although it's important to understand things at a high level, and then not get into mechanism, because if you want to intervene, it's through mechanism. And I want to just introduce the terms that most people will be very familiar with: personalized and precision medicine. Personalized medicine is this person has a particular genome, has particular, in the case of the brain, experience, synaptic weightings, particular set point in terms of affect, depression, potentially, and may have particular anomalies, such as you see in schizophrenia. And that person has to be treated individually and somewhat differently than the other patient with, let's say, depression or schizophrenia is treated. That's the most extreme and remains a hope. But right now, generally, the best we can do is precision medicine. So we can group subsets. And right now, for example, in schizophrenia, we would like to group subsets of schizophrenics based on how they would respond to medication. Who will respond to glutamate-type medications? Who will respond to dopamine-type medications, dopamine blockers? Who will respond to this? Who will respond to that? Or to take another example, a lot of the drugs that have failed spectacularly, such as Vioxx, because it gave people heart attacks. It was an non-steroidal anti-inflammatory drug. It failed because a subset of the population could not metabolize it in the same way, did not metabolize it in the same way, and caused problems for that subset. So precision medicine is pick out the subset that can take this medicine, pick out the subset that can't take this medicine, pick out the subset that will benefit from a particular medicine. So I've already said we're dealing with spatial scales up to the whole body. And when I say we're dealing from temporal scales now, and you will see here we have temporal scales down to the millisecond, which would be action potentials, up to, we should have years here because things development happens over 10 to 20 years, development of the brain, and degeneration happens over 10 to 20 years on the other end of life. But in terms of, for example, just drug treatment, an example would come from Parkinson's disease, where we give Sinemet, and the Sinemet may take effect. Sinemet is a combination of L-dopa and dopamine. It's to be a dopamine agonist to reverse the problems with the degeneration of the substantia nigra, which is altering function in the striatum. It takes effect in a couple hours. So you say, okay, we understand that. We're going to understand that pretty well with these limited temporal scale models. But over the course of 10 years, you find, usually 5 years actually, you find decreasing efficacy. So things are changing, and things are changing in response to the constant bath of dopamine that the body, the brain is seeing. So how are we going to combine machine learning, particularly nowadays deep learning, which is so powerful, and multiscale modeling, mechanistic multiscale modeling? Various ways to consider it. One is that deep learning can itself be used as a direct model of how the brain works, how a neural network works. And in this context, we might use mechanistic modeling to better understand how mechanisms happen in what deep learning tells us about how the brain works. Certainly, all kinds of machine learning, of course, makes an excellent tool for data interpretation. So that includes not only experimental data, but also the in silico data that comes out of the multiscale models, because the multiscale models are experimental objects. They are too complex to encapsulate in a single equation, and you end up running lots of simulations and interpreting them in the way you would interpret a rat slice or a whole rat, which you're recording from. Machine learning can be used as a means of reducing dimensionality of one or more scales to create hybrid models, and that would allow you to then take out a scale potentially. So for example, we always take out the scale, because we can't simulate it, of molecular dynamics. We're going to assume that ion channels behave in a certain way with voltage sensitivity, and we're not going to simulate that. And that would be a very reduced scale. It wasn't developed by machine learning. It was developed by Hodgkin-Huxley. And it's not only the single, obviously not the single channel, it's a population of channels in that case, but we can imagine similar efforts using machine learning or deep learning as an embedded process that would give you the mapping in time that such a reduced model gives. Similarly, mechanistic modeling could be a way, and I think I said that before, of exploring the results from deep learning. How does this come about? What can we say by understanding better the mechanisms of the way neurons are really firing? I think, as we discussed today, I hope we'll find many other applications, and I'll say in a moment where we have our Google Doc. I've just seeded it with these comments here as a way of adding further things for discussion at the end of the day. So we have, though, a mind-brain-body problem. We, of course, want to understand all of these levels. Marr, who most people will be very familiar with, David Marr, came up with the notion that we could understand the brain by understanding the problems that it solves, then going down to an algorithmic level, then going down to an implementation level. And from his perspective, the implementation level really didn't matter, because it could be silicon, and it could be neurons, and it could be anything else. So in paraphrasing him, perhaps somewhat unfairly, we would say he can understand the mind without understanding the brain. And I would point out, as many others have pointed out, that algorithms, the second level of Marr's triad, are non-unique. And for example, no one would have expected that vision, which is what a problem that Marr in particular had interest in, would be broken up into two streams that then have to be bound back together, giving you the binding problem. The binding problem is a problem that doesn't arise until you start to solve the visual problem in this peculiar way, which is evolutionarily makes sense that you have these two needs, where and what. This kind of thinking has been most thoroughly explored, I think, in stomatogastric ganglion by Eve Marder and her colleagues. So down to the cellular level, Goldwash has shown with Eve Marder that the the dynamics of the cell show non-unique solutions in terms of ion channels. And then at the network level, Astrid Prins and Eve Marder again have shown how the solutions there are non-unique. There are many ways of getting to the same thing, and depending on how you get there, you have different mechanisms. And so you'll have different drug treatments and different surgical treatments and different electroceutical treatments. The other point to be made here is Edelman's notion of neural Darwinism. These are systems that are evolving, perhaps, as Edelman suggested, in the individual, as well as, of course, evolving across time in the species. Another point is subsidiarization of neural control. It's always amazing how far a chicken, or as it turns out, a rat or a cat, can run without a brain. And so there's a lot that goes on at lower levels that we rarely think about because we're so neocortex focused and certainly brain focused, whereas the spinal cord and brainstem are really able to do a remarkable amount. And so at every level, you've got things that can be done at that level, and then you have control from higher levels. Fast and slow thinking, of course, vastly popularized by a recent book, fast and slow reflexes, and some very fast responses, remarkably fast. So for example, Thorpe has written about how you can identify scenes in as little as 1 to 200 milliseconds, and points out that a baseball player will be able to interpret where the ball is going within, well, 200 milliseconds is basically all he or she has to hit the ball. So there's really very fast responses that, again, require subsidiarization, require practice, require presumably the cerebellum, and very different systems, sets of systems that are interacting in this complex way. I found a quote recently from Terry Sinyovsky, who will be speaking this afternoon. The body is more complicated than the brain to replicate. So also, when you think about how people work or how animals work, you have to consider the end organ, and the end organ has its own complications that the brain and the spinal cord have to take into account, so that you really have this full system. And of course, you know, in medicine we always talk about holistic medicine. We're treating the whole patient. In terms of understanding, we also have to think somewhat holistically. And another point to be made here is that the stuff that is easy for computers to do, like higher math or now Go and chess, not that that's easy for them, but they do it remarkably well, is hard. Frankly, Go and chess are hard for most humans. I can't play Go at all. I play chess extremely badly. But the things that are easy for animals to do, such as a housefly to fly around and evade capture and mate and feed, are very hard for a supercomputer to do. So I think they're very different algorithms, very different implementations, and those implementations in part are giving rise to very different algorithms that are being used. And I don't think that's by itself a controversial statement. So what are we doing today? Well, we'll start in the morning in 5 minutes with my colleague and co-organizer, Sam Nemotin, who will talk about reinforcement learning in the context of a couple different visuospatial tasks. Then after that, we'll hear from Gunnar Sedersund talking about much higher level thinking about health and treatment of disease with the whole body involved, that point we just made, but here talking about maybe even different systems, so hormonal systems, immune system, all interacting. And eventually one would like to think about your biome and how your gut biome is interacting with your brain. There's a lot of crosstalk, a lot of influences. Then Danilo Chok, I practice this, is going to talk about precision psychiatry, again, precision medicine compared to personalized medicine, where precision is take a group of people and try to understand them separately. Then Daniel Derstowitz, again, a psychiatry talk here, deep learning. Then we have a lunch break. We did have one cancellation, so the lunch break will be a little bit long, which I'm sure no one will object to. And I assumed we'll be running over a little, so I put that at about 1300, 1 o'clock. This is all Eastern U.S. time, and so you have to correct if you're elsewhere. In our afternoon, my afternoon, Thomas Serre will present on circuits to deep learning in vision. So there's this tie-in, tying together some of the circuitry issues, which would be mechanistic, to the deep learning, which would be a machine learning algorithm. After that, Terry Sinyavsky will talk about the temporal dynamics in a particular inhibitory excitatory network in a working memory. And Kenji Doya finally will talk about multiscale data assimilation from the many scales that people do experiments at. Finally, we'll end up with a discussion, and I hope as many people will join in, not just the people who presented. And so for the discussion, Sam and I have set up a Google Doc that will let you add points and questions and potentially additional little chats, pieces of chats you can add to that. And of course, you're not going to copy this down, which is why it says ... I will now copy that address into our chat. So for those who are not familiar with this format, we do have a chat room where we can say little things, and then we have a separate question room where we can ask questions that will be addressed to the speaker or make comments. And so that's two different chats, and then this document in Google will be a third type of chat. So let me hand it over at this point to Sammy Moten. He works at NKI, which is the Nathan Klein Institute, and his title is not readily found by me. So he will tell you his title.
All right, so thanks, Bill. So I'll share the screen now, and hopefully it'll work the right way. Okay, entire screen. So is that all visible now? Screen. Can you hear also?
I hear you. Whoops.
And the screen is, the slides are visible?
I hear you, but I don't think I see your slides. I'm trying to get rid of my sharing. Wait, something came up. Yes, your screen has come up.
Okay, so I'll get started then.
Great.
Okay, thanks. So the topic is using reinforcement learning to train biophysically detailed models of the visual motor cortex to play Atari games. So that's the visual spatial task that Bill was talking about. So just an outline of the topics that I'll go through here. So first, I'll just briefly give an overview of some of the deep reinforcement learning algorithms that are used. And then I'll go to the more biologically plausible reinforcement learning algorithms and show how they can be used to train sensory motor cortex networks to control arms to reach towards targets, stationary targets, and then go on to more dynamic environments in the video game setting where we train visual motor cortex to play the games. So deep reinforcement learning, of course, has very impressive performance. It's state-of-the-art in specific behaviors and computational tasks, including game playing, like Bill mentioned in the game of Go. And it became kind of famous when they trained these deep reinforcement learning networks to play the Atari games using just the pixels, basically, and then a feedback signal. And they've also been used a lot recently for object recognition. So Google image search, for example, uses some of these deep learning algorithms. But they do have certain problems. So one of them is that they require huge huge database of training examples and pretty demanding hardware infrastructure. So it's not always so simple to run these algorithms, and that suggests that they might not be as efficient as they could be. That could be a hardware issue, but it could also be an algorithm issue itself. So there could be room to improve some of these algorithms. And they also use some biologically unproven learning rules, which are somewhat controversial whether they exist in biological systems and in the brain, like error propagation and stochastic gradient descent. So those are the traditional algorithms that have been used for these deep learning algorithms. And another problem is that the networks that they use, they have pretty simplified neurons that don't even include spike time. So that makes it difficult to compare against experimental data that's recorded from animals, for example. And then that will potentially limit insights into how short timescale activity in the brain is contributing to perception and behavior. And this figure over here is showing let me see how to get the pointer, is just showing how the computing power and hardware demands from deep learning algorithms has increased over time and kind of outstripped where they need to be. So some more work on improving algorithms could give more efficient results possibly. So this is a figure that you've seen probably recently from some of the talks here at the CNS meeting. So this is from a paper in 2015 where they trained this deep network to play these Atari games. And so the question that's motivating some of this work is what's missing in these networks and models. So one thing is that the neurons are kind of simplified. They're just these rectified linear units. So they integrate the inputs, but they don't have realistic temporal dynamics, and they don't emit action potentials. So they have less precision in terms of their in terms of their temporal integration and input and output processing. And the circuits tend to be somewhat simplified too, where they generally use feed-forward architectures, which lack recurrent and feedback connectivity, which is kind of a critical feature of the brain, where there's a lot of top-down connectivity important for different computational tasks and so on. And again, the learning algorithms tend to be simplified, which are questionable biologically. So a recent paper was looking at trying to add a little bit of realism to these kinds of architectures for the game playing tasks. So they took an artificial neural network, an ANN, they trained it with gradient descent, and then they converted the neuron units into these spiking neurons. And they had this algorithm for converting the weights so that they would be appropriately scaled. But then the outcome was that there was some degradation in performance, but there were also some benefits in terms of robustness to visual occlusion in the game scenes that are presented to the model. So this is a kind of a hybrid approach. And then on the other side, we have these data-driven biophysical neural circuit models, which have really taken off because there's a lot of data sharing, and they've been developing for many decades. And this is an example of an auditory thalamocortical circuit model, which we've been using to start to investigate the role of brain rhythms in auditory and speech processing. And you can build a lot of rich biological detail into these models. For example, there's many different cell types, many interneuron types, and different types of pyramidal cells, and their connectivity patterns are all determined based on anatomical, electrophysiological, and circuit mapping data. So there's really a lot of complexity in these models, and they're all constrained using this data from experiments. And once you have these detailed circuit models, then that adds computational power compared to the simplified artificial neural networks. So for example, this is a layer 5 pyramidal cell from mouse motor cortex. And this is the reconstruction. So you see these giant dendritic trees, and they can integrate spatiotemporally varying stimuli, much more complicated than what you can process with a point neuron that's typically used in these ANNs. So each neuron ends up being like a network. And another benefit of these models is that they allow you to compare signals to experimental data at perceptual timescale. So this is showing from that auditory model. This is spiking activity from different neurons. This is a raster plot. And then this is current source density from laminar electrode arrays recorded from non-human primates. And then we actually can now simulate this using the same electrode configuration. And then that allows you to compare how the signals in different cortical layers are contributing to input and output and information processing. And another important feature of these kinds of models with the excitatory and inhibitory neurons is that when you have interneurons with different timescales, synapses with different timescales, that contributes to different rhythms. So that's kind of a major theme in these neuronal network simulations, where you have these slow and fast oscillations, and they're coupled in time, and the low frequency can modulate the faster frequency. So that can contribute to different information processing, which is also lacking in the artificial neural networks. So the motivation for this work was to use more realistic microcircuits and cell types to first provide insight into biological dynamics and their contributions to functions, so oscillations, spike time and synchrony, and intracellular dynamics. So by using modeling, you can you can better understand how they contribute to function in this game setting, because the games provide controlled setting to study the contribution of specific dynamic features in learning and behavior. So that was the main motivation for this. And then we want to see if we can use neuroscience to improve our engineering algorithms like the deep learning networks, and that's a kind of more of a long-term goal. So the first example of using this kind of architecture with more detailed neurons is learning these sensory motor transforms. So this was a model from almost 10 years ago, back in 2012. So we built this model of multiple areas, and we were trying to train it to, this is a sensory motor cortex model, and we were trying to train it to control a virtual simulated arm to reach towards a target. And this is a fixed target here. So we used spike timing dependent reinforcement learning rule, and that was which takes a global error signal. So basically, initially, the model is set up with random weights, and then it produces these movements which are kind of stochastic, and that's called motor babbling. And then over time, when the arm moves towards the target, then the synapses which were recently activated get reinforced, and the ones which contribute to the wrong movements are punished, and they get weakened. So over time, this learning rule, spike timing dependent reinforcement learning, can contribute to the proper behavior. So this shows the rule in a little bit more detail. So the spike timing dependent part is when the presynaptic neuron fires, then the postsynaptic neuron fires within a short time window, then that synapse is tagged, and then there's an eligibility trace which is activated, and then later on when the behavior is assessed, so that could be from the dopaminergic reward system, so the reward and punishment signals will reinforce or punish that synapse based on the behavioral outcome. So we could model it in a lot more detail, looking at intracellular mechanisms like calcium-dependent signaling, but this is just kind of a phenomenological model that we're using here. And so it is effective in training that network architecture I showed. So this shows the starting location of the 2D arm, which we're training, and this is the target location. So after a long period of training, so the arm is the network is able to control the arm to reach towards the target, and you can train it on different targets. And then compared to the naive network, this is the error in the movement. So the naive network, which doesn't have any training, stays basically at a high level of error, but the trained model is able to reach towards the target that it was trained for. So that shows kind of a first case that was effective in using this paradigm to train these models. And then once we have the network that was trained, we can look at the dynamics and try to understand what it contributes to function. So this top part here shows a raster plot of the different neural populations and their firing times. And it shows the naive network without training and after training. So there's a lot more synchrony in the population activity. And that could contribute to more effective activation of the muscles, the simulated output from the motor population that controls the movements. And then we also looked at the information transfer between different populations. So that was increasing too. So these circles here are the different populations in the model. So ES is the sensory neurons, EM is the motor excitatory neurons, I is for the interneurons. So before training, the dashed lines represent the information, the strength of the information flowing between the populations, and the solid lines are after training. So there's certain populations increase their information transfer. So for example, the sensory neurons increase the transfer of information to the motor population. So that suggests that the sensory information is being used more effectively to drive the output. And this is another example from a more detailed model, which was developed by our colleagues. And so use this detailed model of motor cortex to train this more detailed musculoskeletal arm model and a robotic arm model in the real world to reach towards a target. And they used different targets. And by using this more detailed circuit model, we can generate hypotheses and predictions on what each cortical layer is contributing in terms of the computations. So that video there showed after training that the arm, both the simulated and the robotic arm, are able to reach towards the target. And so, yeah. So then we wanted to scale up this kind of architecture to a more complex behavior in a more dynamic environment. So that's the video game environment where things are changing and moving around, and you have to use more predictions basically on where the ball is going. So this is the Pong game. So you have two paddles. Probably most of you are familiar with Pong, but the idea is it's basically a 2D version of tennis. So you have to hit the ball to the other side and get it past the opponent, and then you get a point. And so this was an Atari game that was trained with the deep learning architectures that I mentioned in the beginning. But then we wanted to see if we can use the same kind of architecture that I showed in the more realistic neuron models and biologically plausible learning rules. So there are these time-varying inputs that are projected to a thalamic model, for example, that can go to a visual cortex model, which goes to a motor cortex, and then the error signal is used to train the synapses, basically. So this is just a schematic. And then the architecture we ended up building ended up being more complicated. So at the early stages of processing, there's a retinal area, which basically is a topographic arrangement of inputs, and the pixels are when they have a high intensity, so that drives the retinal cells to fire, and that projects to a thalamic area, which has topographic architecture too. And then there's these interneurons there, which provide feedback inhibition, and then the thalamic circuit projects to visual V1. So we modeled it with the different excitatory neurons and interneurons, which take topographic inputs, and then there are these direction selective neurons as well. So each of these patches processes input when there's movement in a specific direction. So by using the topographic arrangement, then we can detect movement directions at any pixel location, basically. And then all this visual information is projected to motor area, which has specific motor populations which produce movements in specific directions. So the paddle is going to go either up, down, or stationary. And all of these synapses between the visual areas and the motor areas are tuned through learning using the spike time independent reinforcement learning rule, which is loosely based on the dopaminergic reward system. And the neurons in this model are simplified compartmental models. So they have spike times, they have ion channels, and they have more realistic synapses, which allows more realistic dynamics to emerge compared to the commonly used deep learning algorithms. So one of the first stages in the processing is that we have to do this image processing to extract the movement directions. So we do a thresholding on the input image, which is from the game environment. And after that, we find the object, basically, by thresholding the image and then getting the pixels with high intensity and then finding their boundaries. And then in successive frames, you can calculate the movement directions. And then at each pixel, you have a movement direction. So that's shown here. This is an untrained network. It's just showing that the movement directions are detected accurately. And you could also use other optical flow algorithms, and that will work pretty well too. But this gives a little bit more precision to the pixel, to where the movements are occurring. And then for the reward paradigm, so there's different ways you can implement reward and punishment in the model. So you can either use just the points that the Atari game gives you, and that kind of reward works, but it's a little bit more sparse in terms of how often it occurs in time. And so it could take longer to train these models. So to allow the models to learn more quickly and to accelerate the learning process, we used intermediate rewards. So we provide rewards when the paddle moves towards the ball, for example, or towards the location where it'll intercept the paddle's Y location, where it'll cross the right side of the screen, basically. So that will be where, when the paddle moves towards that target location, then we'll give a reward for the synapses which were recently activated. And when it moves away from it, then it'll get punished. And we can also use other intermediate rewards, and so I'll explain that a little bit more in detail here. So this shows the target trajectory reward paradigm. So this shows the ball going to the left. That's the AI player, and this red location here is where the ball will intercept on the right side. So we can train the model to move towards that location dynamically. And that will allow the learning to accelerate more quickly and to take less time to train the full model. And okay, so another reward paradigm we use is the hit-miss paradigm. So when the ball comes close to the paddle, if the paddle moves away from the ball, and the opponent ends up getting the score, then we punish the model. But if the ball hits the ball, then you can reward it. And sometimes it takes a few volleys to get a point. So that's why we consider it an intermediate reward. So it doesn't necessarily lead to an official point in the Atari game, but this could allow the model to learn intermediate signals. So we call this teacher-based reinforcement learning, because we're instrumenting the model to have more intermediate rewards, which is kind of like a guided system. And then after a lot of training, then we can look at the performance. So this shows the learning curve for these different measures from the model that I was describing. So this is the follow target score basically over time. So over a long period of training, the performance improves a lot, and also the hit miss score improves a lot. And so that's when the ball comes to the paddle and it hits it versus moving away from it. So that score is improving a lot too. And the score miss is how many points basically compared to the opponent is our model getting. So it's improving too over time. And we probably need to run it for a lot longer in order to win, but this is just showing a proof of concept that the system basically works. And another thing which is an issue in these more detailed models is that if you just let the weights grow continuously, then you're going to have problems with hyperexcitability and epileptic activity. So we had to implement this weight normalization rule, where every so often the weights are scaled down. So the pattern of weights is preserved, but the average weight is just set down. So that's shown here, that the weights are increasing. This is without normalization. This is with normalization. So periodically, the weights are just scaled down to prevent this hyperexcitability. And we also found that the hyperexcitability is degrading the performance. So similar in epilepsy, you can see problems in motor control and other cognitive deficits sometimes. So this is showing the learning rates basically when we normalize the weights. This is the red trace, so the performance is improving substantially, and versus when we don't normalize the weights, so then the performance is a lot lower. And if we run it for a lot longer, then you'll see bigger differences too, because the epilepsy and hyperexcitability will develop, and then the neurons won't fire properly or be able to process the sensory information properly. But there's still an issue of how often to schedule this weight normalization. It has to be at the right timing, because if you do it too frequently, the weights will decrease and the learning won't set into the network properly. So another thing that we saw, that once we have the training done, then we can look at the weights for specific populations. So these different colored lines here show the average weights to specific neurons. And I mentioned before that there were 3 populations of neurons in this architecture. So we call them the left population, the right population, and the stay population. So they contribute to, the up, down, and stay population. So the up population moves the paddle up, down population moves the paddle down, and the stay population holds the paddle still. And what we found was that the weights to the up and down populations were increasing faster and to higher levels than the stay populations. And that's because the stabilizing population only is active when the paddle happens to be at the exact location where the paddle needs to be and where the ball is moving towards it. So it's still a useful population in command to prevent the paddle from moving away from the target location, but it's less likely that it'll happen to be in the right location. And then the firing rates of those populations are reflecting the weights. And this shows the membrane potential of different neurons in the raster plot. So you see these kind of synchronous patterns of activation. And up here is the motor populations, which also show some synchronous activation too, which could be useful for driving the activation. So that's just to contrast to the more traditional deep learning algorithms where you can't measure these features and then you can't compare them to the biological data in these different tasks. And now I'll show videos of the trained model. So on the left is the AI player, on the right is the trained model. So this just shows a segment of the activity where the model was performing well, and this and it hits the ball and it scores a point, and then it after a few volleys it also is returning the ball. And this shows the activation in different cortical layers and populations. So this is the retinal populations. Here you can see brief activation with topographic information, which is then projected to the excitatory neurons in V1, which have the topographic arrangement. And then this shows the direction-selected neurons. These are the movement directions detected, which closely matches the movements in the video. And then over here is shown the different motor populations. This is the up motor population, the down motor population, and the stay motor population. And we also have an interneuron population there, which is providing feedback inhibition to the motor population to prevent runaway and epileptic kind of activity. So just showing that you can measure the activity at a fine-grained temporal scale over short intervals. So this is 20 millisecond binning to generate these activity plots, basically. And you can show, and it just demonstrates in action that the model can produce the right behaviors after training. So in conclusion, we can use these closed loop models of the sensory, motor, and visual cortex to learn behaviors in the dynamic environments, just using these spike time independent reinforcement learning rules, which are more plausible from a biological perspective. And we're also able to use intermediate teacher-guided rewards to accelerate the learning. So in the long run, these kinds of models could provide potential alternatives to the commonly used deep learning algorithms, and additionally, they offer insight into the biological processes. And when we build in more of the biological detail, so this was a proof of concept, basically, that we can train these kinds of biophysical circuit models to perform appropriate behaviors. And when we add more realistic circuits, so for example, the kinds of models we were developing for the auditory system, we could test specific parts of the circuit and see how they contribute to behavior in the thalamocortical system. And eventually we want to look at human and non-human primate and other animal data during behavioral tasks and see how specific circuit elements contribute to the behavior. So just wanted to acknowledge the funding. So this work was made possible by the Army Research Office primarily for this work, and some of the circuit modeling for the auditory system was from NIH grant. And I wanted to thank all the people who contributed to this work in different ways. There were a lot of people in different times, so thank you. And here's the bibliography.
Thank you, Sam. So we have a couple of questions here. How much time does the training take and what hardware?
So we're able to run the training on a standard workstation, which has, well, it's kind of a high power workstation. What we could use is about 30 cores or so. And it takes a couple of days to run when we use, when we parallelize the model. So that the models were created in the NetPine platform with the Neuron simulator. So that allows you to parallelize the models pretty easily. And then, yeah, so it takes a couple of days at least to get the results with 30 cores.
Does the training include functional plasticity, changing synapse weights, as well as structural plasticity, adding and removing synapses?
So right now we don't have structural plasticity, but we start out with a pretty high connection density, and we randomize the initial weights with pretty high variance. That ends up helping. And so because of the high variance, some of the synapses end up being not useful, so they can get basically set to close to 0 weights. So that's similar to structural plasticity, and it's simpler to model than adding synapses and removing synapses during the simulation. But that would be interesting to try later on.
Okay, another question was whether you have a potential for really a direct comparison with the other successful program, the deep learning program of, I don't know how to pronounce it, NI et al. 2015?
Yeah, so there's a lot of source code online which we're planning to use to compare the performance. Well, right now the performance isn't at the level of the deep learning algorithms, and one of the reasons for that is that we don't have the hardware infrastructure that the people who ran those algorithms have, and we are running these models pretty much continuously, so they are improving, but we have to run them for a lot longer probably and work out some some details of the algorithm to get a little bit better performance before we compare. But we would like to compare to those deep learning algorithms, yes, for sure.
Great, thank you very much. So our next speaker is Gunnar Cedersund from Linköping University in Sweden, and he will speak on the topic of multi-level modeling of the brain, from intracellular signaling and neurovascular coupling to whole body crosstalk and clinical implementations. Gunnar, are you able to share your screen there?
Yeah, can you hear me now?
I hear you.
Do I need...
Yes.
Okay, very good. So I don't know where is the share screen? I didn't find it yet.
Let's see if I can find it. I think you have to hover over something, probably over yourself. Is that right, Sam?
Hover over myself. Maybe here. Let's see. Share screen.
Okay, good.
Let's see. Is it working? Do you see my screen now?
I see you are screen sharing. Sam, did you release your screen?
Yeah, let me try that. Maybe I'll shut my video to make sure that it stops my sharing. Let me see.
Okay, so let's try again, Gunnar.
Okay. And yeah, now something else happened. Let's see. Share screen. Allow. Oh no, it doesn't work. I'm using the wrong. Wrong.
You have to click on the when you start with share screen, you then have to click on your picture and then click on share. So
yeah, I did it, but it seems like you can't use Firefox, and I'm using Firefox. I'm terribly sorry about this.
Odd. Anyone been in one of these meetings before who knows how to do it? They
Can't
can
give access to
come on the chat, or if Sam, do you know?
Okay, let's open the same in another. Copy.
Hi. So you're having trouble with the sharing screen now still?
No.
Or?
Should use this instead. Let's see.
Let me see. So it might be a Chrome issue someone's suggesting.
Well, he said he was on Firefox. I would have thought it would work with the major browsers. Oh, I think we did have one more question for you, Sam, actually, that I didn't see in time, so I'll present that to you meanwhile. Do you add stochasticity in the neuron types for the decision making? Whoops. Did you hear that, Sam? Are you muted or am I muted? Sam is muted, apparently. Okay, I unmuted you, Sam.
Oh, okay. Yeah, so right now there's no stochasticity in the output control. But there is an option that we have for adding noise to the synapses, which we haven't found too effective right now. I mean, there's randomness in the initial wiring of the network, so that contributes to some kind of stochasticity in terms of the behavior and dynamics, but not explicit randomness in terms of the motor output encoding.
Okay, so I have Gunnar back. Sam, now you can mute yourself, I think. And Gunnar, I'm now seeing your screen, I think, but I'm not hearing you or seeing you. Should we consider moving to a different talk and then coming back to Gunnar?
Yeah.
Who's the next person? Is Danilo Chuk available? Actually, he wouldn't be able to answer me because I haven't put him on, so I guess I can put him on.
Oh, there's Gunnar again.
Oh, we might have him.
Must have had.
Are you having any luck, Gunnar? Should we save you for
Maybe.
one, and should we save you for one and then come back to you?
Yeah, maybe. Can you hear me now?
Yeah, I hear you, but I don't see your screen.
But you saw my screen before.
Before I saw the screen, but I didn't hear you.
This is so weird.
So, is there a way to focus on him now, on Gunnar? Oh, Danilo.
Okay. So, do we want to let's have Danilo speak and then come back to you, Gunnar?
Yeah.
Is that okay?
Yeah, that's perfectly fine.
Okay. So, Danilo Chuk speaking on algorithmic analytics towards precision psychiatry.
Yeah.
Now,
So,
let's see if Danilo
you
can share
unshare
screen.
some of the other screens?
Now, you have to
If I
unshare.
click here, there's 4 simultaneous screens and says I can't do sharing. So
Don't even know
one
if
of you
I'm
needs to
sharing.
log off, it appears.
Yeah, so if you, Bill, if you close Gunnar, then that should work. And I'll close myself too.
Don't do that, Sam, because I depend on you. Okay, so now is that any better, Danilo?
Okay, can everybody see my screen?
Yes.
Yes,
Progress.
changing. Okay, perfect. So what's the timeline? 30 including questions or without questions?
Including questions, yes.
Okay.
But it's okay. It's okay.
All
Go
right.
on.
So my name is Dino Stock. I'm a medical doctor by training with a dual background in systems neuroscience and machine learning statistics. And currently serving as associate professor at McGill's Faculty of Medicine. And we are bridging the activities there with emerging machine learning solutions through my second affiliation, which is Mila Quebec AI Institute. And so the following, what I will try to do is to provide a bird's eye view over a landscape of quantitative modeling solutions and how they relate to the goals and ambitions that we have in our path towards precision psychiatry. One of my favorite slides is this one here, which tries to portray some of the important statistical developments over the last decades. And it is compelling to be cognizant of the fact that most of the quantitative technologies that we are using on an everyday basis in brain imaging, they actually emerged in what is blue here before Second World War, before electronic calculators were invented. And this is really things like t-tests, ANOVA, chi-square, Pearson correlation, and null hypothesis testing with p-values. So it was really after Second World War, in the second part of the 20th century, that a lot of the methodologies that we today may call machine learning, deep learning, for example, that those emerged. And that's things like cross-validation procedures, which provide a different but similar approach to deriving rigorous conclusions from data, which is not equivalent to null hypothesis testing. But also things like penalized linear regression, or in high dimensions, as some people call it, linear aggression. Bootstrap emerged as a very universal tool to assess statistical accuracy in a lot of different modeling settings, up to all the way to random forest, which emerged only in 2001, and the rise of deep learning, perhaps around 2010. So one way to summarize this is the biggest difference between pre- and post-war statistical practice is maybe the degree of automation, up to a point where today almost all topics in the 21st century are very much computer dependent. So what these changes, I alluded to this before, in the quantitative toolkits that we use, it doesn't only change kind of what programs we do. It may actually change something more fundamental on an epistemological level, and that is what types of rigorous conclusions we can actually make based on data. So what you see here is simulation results from a so-called fake data simulation study, where we looked at gene expression type of data. And on the left, you see a bunch of gene expressions that are analyzed with classical statistics, the most common way to generate p-values on this type of data. And on the y-axis, you see which genes come up as relevant. If we analyze the exact same data with a tool that is maybe prototypical for the machine learning community, random forests, that's in panel B, then you see that some of the genes are the same, but they are not exactly the same importances as we see in panel A. So if we overlap this in panel C, then we can see that classical statistical inference based on p-values and a more prediction-based cross-validation estimate type of conclusion. Here, exemplified by the random forest, they do not give you the same type of answer. And so the kind of intermediate summary up to this point is, yes, we went through an increasing automation towards tools with always more parameters that take always longer to compute, as we heard in previous talks. But they may also become always more inscrutable because there's so many model parameters. Our notions of interpretability are likely to change. And the categories of conclusions may be non-identical to the ones that we made in the 20th century, where we were very focused on activities revolving around attaching p-values to single input variables. So if we take this to a broader level, then what you see here is more than 100,000 different potential data sets that we generated by systematically varying a lot of the factors that we typically care about in everyday data analysis. That's how much correlation do we observe between a set of input variables? How much noise do we have? How many variables are actually relevant to predict a certain outcome? How many data points do we have given the number of observation variables, and so on and so forth. So we systematically varied all these factors of 100,000 data sets where we knew the target prediction. And what we see on the left here is each dot is really what is the best possible p-value that you could get on a given data set, and what is the best out-of-sample prediction accuracy on the y-axis for that same data set. So the point is, there's a non-identical relationship between the most common ways to do null hypothesis testing and p-values in linear predictive models here, and using a same modeling solution to try to generalize to new data points drawn from the same distribution, which you see on the y-axis. So we can zoom in into a space that we typically care about based on p-values that's on the right side here. And you can easily see that it is much easier across all these data sets and data analysis scenarios to obtain at least one statistically significant result. But these same models showed a drastic variation in how well they explain variation in new data points drawn from the same distribution. So if you put it on its head, it is much easier to get statistically significant results, but they're not always predictive. Whereas if you establish predictive relationships, such as by cross-validation type of procedures in a linear model class, then those most of the time will also give you statistically significant results. So if you now think that all these were just made up data and we tweaked it in some way, here's just a very quick slide from 4 common data sets from textbook statistics sources, data sets that have been analyzed again and again over the last 20 to 30 years. And we show that across common data sets from the biomedical space, that all combinations of statistically significant and successful prediction are possible, right? So in a given data set, most of the time in a given research project, we usually just have one data set. And so in a given data set, all combinations of finding significance but not predictability but not significance, and so on, are possible. So in the following, and that's the second of 3 parts in this talk, I will come in and say that I will make the argument that classical null hypothesis testing, as we have been practicing in the 20th century, and which was an important ingredient for 20th century evidence-based medicine, and it's responsible for a lot of forward progress in medicine, including psychiatry and neurology. In the following, I'll make 10 different arguments why establishing p-values in significant relationships is probably misaligned with many of the goals that we have in precision psychiatry and computational psychiatry. So one first result citation is this one here from the 70s, actually, a philosopher. So inferences but not prediction explain what has happened, and predictions but not inferences forecast what will happen. So in my own words, this kind of means that, which is sometimes forgotten, that p-value statistical significance type of inferences they very much rely on a criterion of optimality that is not directly targeting prediction performance in future or other data sets. So if you really want to put your finger on it, then p-values null hypothesis test active type of methodology in direct comparison to cross-validated machine learning types of predictions. And this takes us back to the historical overview that I tried to portray at the beginning. So a lot of the questions that we are asking more and more, now that we have internet scale data, now that we have the computational power to run models with millions of parameters, so it really just recently became possible to fit models and fit them well up to a point where we can obtain accurate predictions about quantities that are relevant in the future, including, for example, symptom prediction from drugs, drug repurposing strategies, or treatment response prediction in psychiatry. So another aspect to emphasize, again, that inference prediction are just completely different modeling paradigms in a lot of the times. On the left, you see some of the general and theoretical properties of classical statistics, such as exemplified by ANOVA, p-values, linear regression, and so forth. And an important foundation of these quantitative tools is really notions like degrees of freedom, asymptomatic consistency, right? A lot of the theoretical proofs, they very much rely on taking sample sizes to infinity theoretically. And this leads to kind of like invalidations of differential processes like double dipping, circuit analysis, and so on. And what I like to emphasize, however, is that the predictive paradigm, such as exemplified by many of the tools and modeling regimes in the machine learning community, it has a completely different basis in terms of theoretical pillars that really justify and define the boundaries of these modeling paradigms. So those are things like Vopnik-Shavon-Kiss dimensions, but also probably approximately correct learning, cursive dimensionality, notions of hypothesis space. So how large is the space of modeling solutions that are potential results of a particular analysis that the investigator conducts. And rather than doing a lot of mathematical proofs where the sample size is taken to infinity, there's a relatively larger focus and the theoretical work in this community on proofs that show guarantees as a function of increasing sample size. So long story short, the theoretical foundations of what justifies now hypothesis testing, things like t-test and ANOVA and so forth, they mostly emerged before Second World War. Whereas the things that I mentioned here on the right, Vopnik-Shavon-Kiss dimensions, but also what's not shown, probably approximately correct learning, different theoretical frameworks, they very much emerged in the middle of the second half of the 20th century, so more like in the 70s and 80s. So the summary from this slide is, again, that making accurate predictions about single variables and potential mechanistic insight is just a different modeling agenda than trying to merely show useful, accurate predictions in new data. And that is also reflected in the theoretical foundations of these different modeling paradigms. Here's another quote. And so this is really from one of the classic textbooks in classical statistical inference. And it's really on page like 300 or 400 that they say a type of inference that we have not discussed until now is prediction. So of unobserved random variables, this is the type of inference that they didn't really consider up to that point. So you can look at kind of thick, recognized brick textbooks that just do hardly discuss prediction to new data points. And there's a lot of reasons why this made sense in this modeling paradigm. So here's the second of 10 arguments. Once you have a p-value, it is not immediately possible to take this single quantity, go to new data points or new patients or new brain scans or new genetic microarrays, and to make statements about a new data point or person that you have not observed yet, right? So one way to kind of understand that is that what you see on the left, null hypothesis testing, it very much kind of uses up all your data, all the data points that you have to really come to the conclusion whether or not a particular input variable, like factor, covariate, whatever, has a relevant relationship to a given outcome, such as or not. In cross-validation and other resampling schemes, which you see on the right, there's a different logic to it where models are fitted in sequence to different subsets of the data. So you have a lot of different KNet models, and you leave out a certain part of the data points drawn from the standard distribution that are independent from the previous ones. And this is really what allows you to get an accurate, nearly unbiased estimate of the expected prediction performance in new data points, microarrays, patients that you may observe in the future. So here's a third point. So closely related to the previous one, only if you can apply a model to a single data point, it is also possible to, for example, design a model this year, but then deploy it next year in 2021, when a new patient enters the practice, shows his or her genetics, behaviors, blood profile, and so forth, and to really use that model that we built in the past to derive conclusions in the future. Another point that is made every once in a while is that we know since at least the 30s of the last century that p-values null hypothesis testing were not designed to scale to very large data sets. So what you see here is, as the sample size increases, a Pearson correlation coefficient is one way to do null hypothesis testing. It actually becomes significant for always smaller Pearson correlation coefficients. So one example is, do you have this here? One example is the UK Biobank study, one of the early ones, in 10,000 people. At this sample size already, most Pearson correlation coefficients around 0.1, which most of us are taught at university at school, are not that strong. They're all statistically significant, even after correcting for multiple comparisons. So if you say it in a different way, if you have thousands of data points, which we more and more often do, which we probably will require to approach a future of precision medicine, it will be always harder not to obtain statistically significant results. So here's another way to put it. It's commonplace among statisticians that chi-square and p-values can be viewed as a crude measure of sample size. This can be framed as a distinction between practical and statistical significance, right? So it's easy to get statistical significance, large data sets, but it's harder to get practical significance. Well, let's skip this one. Here's another one, 0.5. I think that is less often mentioned from what I see. So classical p-values, null hypothesis testing, inferential statistics, as we practice in the 20th century, they have not been made for the high P setting. That means if you fit some model that appreciates thousands, tens of thousands of parameters, that's what we do in polygenic risk scores. That's what we do in what the brain imaging community calls multivariate pattern analysis. P-values have not been made for that. And here in this book, people say, we have seen that in P bigger N, more variables and observations, it's easy to obtain a user's model that has zero residuals And their conclusion is that squared errors, p-values, r-square, and so forth should never be used on the training data as evidence for a good model fit. Another one is that many of the classical statistical procedures, including ANOVA, they rely on dichotomic thinking. So for example, what we want more and more in computational psychiatry is really to compare many different disease categories at the same time. So not only what's the difference between healthy and schizophrenic individuals, but what's the difference between healthy, schizophrenic, bipolar, depression, ADHD, and so on and so forth. But the thing is that if you look at how ANOVA, classic ANOVA, actually implements such kind of multi-outcome setting, then you realize that there's often an omnibus type of test where you say, okay, is any of my target groups different from any of the other groups? Then I obtain statistical significance. So there's no technical distinction between whether kind of schizophrenics are different from everybody else, or every disease is different from any other one. So another one related to the previous point is multi-outcome modeling. So the machine learning community just has so many more tools, and historically a much stronger emphasis on modeling paradigms where you consider several dimensions of variation at the same time. So instead of on the left comparing schizophrenia and healthy or ADHD versus healthy, is more possible to just compare dozens of different lifestyle markers or aspects of disease or different facets of a clinical outcome of interest. So there's just a richer toolkit to do that. Also, what we see more and more is convenience samples, open data sets, electronic health records, other data sets that we just happen to have and that we're not, like we do still most of the time, carefully collected in a previously planned, well hypothetically motivated study of a few subjects invited to the local laboratory. So more and more often, we want to combine different sources of data that we happen to have. And this will more and more include, for example, sensor data, for example. And that really raises the question, how should we actually combine all these different, very kind of like heterogeneous in nature data sets? And again, the machine learning community has probably more tools on the tool belt to integrate high-dimensional sources of variation into a single model estimation than many of the tools that emerge in the classic statistics community. 0.9 out of 10 is similar to the previous one. If we analyze more and more sensor data, open data sets, or reanalyze data that were acquired for a different purpose, oftentimes we will look at observational data rather than controlled data. And there's a lot of implications that make it harder to really make careful statements about disease and brain biology in these much more messy and much more heterogeneous data sets that were not perhaps acquired with the same rigor or that have much more missing data and so on and so forth. So the last point is the whole space of latent factor discovery. So when we talk about disease subtyping, intermediate phenotypes in genetics, brain imaging, and other areas, I think it has a very close relation to latent factor modeling. And again, I think the machine learning community, I mean, we have PCA since 1900, and ICA emerged in the mid-90s. But since then, in the last 20 years, a lot happened in the latent factor modeling space. And the machine learning community has kind of generated many of the recent inventions that lend themselves more directly to disease subtyping. So 2 examples from my lab as a third part of this talk, just very quick. The first one is this year. So we heard a lot of talking about deep learning and its relation to psychiatry and brain imaging. So what we started 2 years ago is what should still be the most comprehensive assessment of the feasibility and potential hard advantages of deep learning solutions in brain imaging MRI in the UK Biobank. So as many of you know, the UK Biobank is the largest existing open biomedical data set at this point. And so we benchmarked how do always more complicated models that have always better theoretical potential to yield better predictions, how do they actually scale with always more subjects? That's on the right, in comparison to reference data sets from the machine learning community. So very quick, what you see is that on the left, an MNIST 10 class digit classification, which is one of these favorite goals that the machine learning community likes to use to benchmark models. You see that as you have always more data points, linear models are always more outperformed by kernel methods in green, which are more elaborate, capture more complicated relationships. And the kernel models, in turn, are always more outperformed by deep learning models in blue. And that's also the case in the fashion data set on the bottom left, which has a more complicated classification problem. If we now turn to brain imaging data and the UK Biobank, as I mentioned, the top right, this is structural brain data from an atlas. And on the bottom right, you have functional MRI data, so functional connectivity edges in thousands of people. And you see that not only deep learning does perform as well as kernel methods, it goes beyond that. Deep learning models cannot be reliably outperformed by linear models. So I'm talking about penalized linear regression, linear support vector machines, logistic regression. We do not see in 10,000 people evidence that deep learning can capture exploitable nonlinear structure. So that doesn't mean that deep learning is necessarily useless for phenotype prediction and brain imaging data, including disease classification. But if the main goal is to achieve the best possible prediction accuracy, we find little evidence that brain imaging has the granularity, has the richness of complicated intervariable relationships to really make distinguishably better predictions compared to even linear models. So the last example is from my lab, just this one here, just one slide. So what can we do with machine learning, though, in brain imaging in relation to psychiatry? And what we did here is we tried to ask the question, which types of experiments, which cognitive paradigms that we could administer to healthy subjects and patients with a diagnosis of schizophrenia are actually most promising if we're using brain imaging. So what you see here is the result of stacking analysis in a European across-site data set, 500 people, where we rank in a data-driven fashion which classes of cognitive functions yield most reliable differences between healthy and schizophrenic individuals. And in the top 10, you see a lot of topics, brain systems, neurocognitive systems that we have been identifying and investigating over the last decades. But there's also several of them. Yeah, I'm done. So conclusions. I think that's most important to me personally is, so different statistical regimes serve distinct research goals. That's one. And that shouldn't be a matter of tradition, habit, or taste. That's my main conclusion. I thank my collaborators for the inspiration on the left, and I thank these institutions on the right for the financial support. I'm happy to answer any questions. Any questions? Any questions? Okay, stop sharing. Okay, stop sharing.
Okay, now you can hear. Okay, so thanks for the great talk. Yeah, so we had a couple of questions. The first one, is there a process for taking old data, doing a meta-analysis that would let you divide up the population into subpopulations for precision medicine for these subgroups?
Yeah, I'm not exactly clear what the exact question is.
I'm not exactly clear what the exact
I think one kind of warning that I could give from what I see in computational psychiatry and imaging neuroscience approaches to computational psychiatry is that the more we combine different data sets and different populations and different sites, the lower our prediction accuracy. That's just empirically what we observe, right? So all of us would like it if we combine always different data sets and then our modeling gets better and our prediction gets always better. Unfortunately, for now, for the most part, that's not what happened. It is very difficult to account for what some people call batch effects. So different sites that use different scanners, for example, or population differences in the types of people that are recruited, different comorbidities and so forth. Handling these divergences and sources of heterogeneity on a technical and biological level of different data sets, different sites, is a major challenge. If we can get rid of those, yes, then it's great. Then we can do meta-analyses. Other questions?
Let me see. Do people in the audience know where they can ask questions? See that little button at the bottom that says ask a question, in case the audience didn't notice that. But let me see if there's any. Not right now.
Okay.
So thanks, and so now we'll go to Gunnar.
It seems like I'm still having the same, or I changed the browser, but it seems like this browser doesn't work either. So keep moving on in the schedule if possible.
Okay, so let me see who's next then. Daniel Derstowitz. Let me see. Let me invite him on screen. Okay, I invited Daniel. Let me see where. Yeah, Gunnar, are you using a Mac? Yeah, there's some settings that you have to change for a Mac. Maybe. Okay, so we'll have Daniel now, and then after we fix the issues with the screen sharing for Gunnar, we'll continue with that. There's some comments in the audience from people who might be able to help with that, and I'll try to look up that information too now. Okay, so Daniel Derstowitz from CIMH Mannheim and the Bernstein Center will now present. Daniel, do you have a screen share set up?
Yeah, but it doesn't show the window, the application window that I need. So it's the same issue we had before. Okay, let me try something. I'll try. Let me briefly try something out here. Now okay, yeah. Okay, can you see my slides?
Yeah, now we can see them.
Okay. Okay, so thanks for joining. I hope you can all hear me and see my slides. I'm going to talk about our approaches for inferring dynamical systems from data using deep learning approaches. So speaking at a computational neuroscience conference, I assume that you all know what a dynamic system is basically, but still want to remind you of the importance of dynamic systems theory in neuroscience and psychiatry first. As many of you will know, it really has a long tradition in theoretical and computational neuroscience.
Oh, sorry. Now we can't see the slides anymore.
You can't see the slides anymore. Okay. How about now?
No.
Okay. I'll go back to the screen sharing here. It keeps, well, okay. Now?
Yeah, now we can see.
Okay, yeah, don't know what's going on here, but okay, let's hope for the best. So just wanted to make the point that dynamic system theory really has a long tradition in computational and theoretical neuroscience, dating back at least to the work of John Hopfield, but likely before, who, as many of you might know, conceived memory patterns as fixed point attractors and the process of memory retrieval as convergence to one of these fixed point attractors. There's a lot of empirical indications of many different types of dynamic systems phenomena. It really has taken off, I think, in the last 5 to 10 years, and you can find many examples. This is just one from the Churchland lab, where they showed these kind of limit cycles which correlate with different movement patterns. But it doesn't have to be all fixed points or limit cycles. You find all kinds of stuff or evidence for all kinds of dynamic phenomena in the brain. So this is here a bit more elaborated idea by Rabinovich and others who thought about these, what they called heteroclinic channels, where you have these chains of cell nodes connected by heteroclinic orbits, and the whole object works like an attractor by pulling in trajectories from the vicinity. And that's a very flexible arrangement for processing sequences, for instance. And that is compatible with many observations, for instance, of metastable activity. So this is one of the results many years ago from our own group, Emily Balaguer at the time, collaboration with Chris Leppish and Jeremy Siemens. We looked at this multiple item working memory task, details not important here, and found, for instance, that you have this typical pattern that you would expect for a heteroclinic orbit with trajectories moving in fast and then slowing down and then progressing fast again. So I don't really want to dive into much detail here. I just want to make the point that cognitive processes are commonly thought of being implemented in terms of system dynamics in theoretical neuroscience. But the point is that we do not have direct access, of course, to the generating equations. We do have models, but flow fields are not directly accessible. At best, we can reconstruct trajectories. So dynamic system theory, as we have argued for a while, but most recently here together with Quentin Huy's in a recent review, might also play a fundamental role in understanding psychiatric conditions. Some things you can easily think of, if you have certain disturbances in the system dynamics, you would expect certain symptoms. For instance, if you have overly strong attractor states, as indicated by this cartoon here of a potential landscape, then if you are in an auditory area, you might get stuff like tinnitus. If you have an auditory percept that you cannot pull out of the attractor state anymore. If you move to frontal areas, you might get something like rumination, these ongoing sequences of thoughts. In depression, for instance, you might expand compulsivity by similar dynamic mechanisms. Motor stereotypies are pretty common in psychiatric conditions, or changes in cortical oscillations. At the other extreme, if you have too weak attractor landscapes, for instance, you might get incoherent thought as you cannot hold on to a mental concept anymore. In the extreme case, you might get hallucinations as representations tend to pop out spontaneously. Attention deficits, obviously. Another important point, I think, are bifurcations. Bifurcations, I think, have been pretty well established by now in epilepsy, but they might also, they are probably a likely explanation in my mind, for instance, the sudden onset or offset of symptoms. I also think it's important to note that dynamic systems theory can serve as sort of a hub, as a central layer of convergence, in the sense that many different changes at the biophysical level might give rise to very similar changes in attractor dynamics, and in turn, similar changes in attractor dynamics in very different brain areas, as I just tried to illustrate in some examples, might give rise to very different symptoms. So that's important, or very important in our minds, because it might have fundamental implications for treatment, because it basically tells you what you need to do is to change the system dynamics. And that doesn't necessarily entail that you need to sort of reverse the same biophysical properties that gave rise to the change in system dynamics in the first place. It might also explain comorbidities, because if you have a general systems dynamical change in the brain, then it might lead to a number of different symptoms depending on the brain areas affected. So now let me move on. So this is basically just a brief introduction why we think this is very important. So what we want to do is identify dynamic systems directly from time series observations. And what we want to use, our backbone are recurrent neural networks, as they are used in machine learning. They are by now, I think, the standard tool in machine learning for processing any type of time series information. So for instance, in language, natural language processing, or in monitoring video streams. So for instance, if you have Alexa or a Siri, that is usually recurrent neural network behind it. For us, it's important that these are dynamically universal. So you can approximate arbitrarily closely the flow field of any other dynamic system. And that is based on two ingredients, the universal approximation theorem, dating back to, say, Benko and Funahashi, and then you can reshape that as a recurrent neural network. So the type of system we are working with are these so-called piecewise linear recurrent neural network. ZT is my neural state vector, A is a matrix of autoregression weights, W is a connectivity matrix, phi is my rectified linear unit, that is my piecewise linear transfer function. And this here, C is a weight matrix for external inputs, and H is bias down. So why are we using that? You might argue that that there's at least a sort of modest physiological motivation. So in our hands, we often found that, for instance, input-output curves of layer 5 pyramidal cells or fast-spiking interneurons in a physiological regime are pretty linear. And also this whole thing here has sort of an interpretation of a neural population model. More importantly, many properties in these systems can be accessed analytically. So you can compute fixed points, limit cycles, and their stability, for instance. Moreover, you can translate. So this is a discrete time dynamical system, as recurrent neural networks in machine learning commonly are. But they can easily be transferred, or not easily, but under many conditions, we can transfer them in ODE systems. That is, in ordinary differential equation systems, which come with flow fields and a lot of analysis tools that make it easier to analyze the system. And perhaps one of the most important points is these allow for highly efficient inference. So one of the caveats, though, with these recurrent neural networks, as I just illustrate them in this vanilla form, is that they have a problem with the so-called exploding vanishing gradient during training. So this is one of the machine learning benchmarks that has often been used to probe that specific problem, the so-called addition problem. dating back to a paper by Hochreiter and Schmidt-Huber. So the idea is you have two series of inputs to your recurrent neural network. One is just random numbers between 0 and 1, and the other are indicator bits. And the job of the network is to spit out in the end the sum of those random numbers that have been highlighted by the indicator bits. Now, of course, the network doesn't know it. It has to learn that by training, and you can make the problem challenging by increasing the time lag between the different numbers that need to be added up. So the classical solution proposed by Hochreiter and Schmidt-Huber, and still one of the gold standards in the field, are these so-called long short-term memory networks. They are sort of engineered systems to solve the problem. They have these linear memory units which can hang on to any state that you basically pump in there. And then they have these controlled input gates and output gates to let information flow in or out. Now, what we want to do, because we are interested in dynamic systems reconstruction, we wanted to keep the simplicity of these, the mathematical simplicity of the recurrent neural networks, the vanilla form of these, but still want to sort of solve that problem. And for that, we basically changed, we added certain regularization terms to the optimization function. So first, as a reminder, A is a diagonal matrix. And now if you briefly blend out these second terms, then obviously if this would be an identity matrix, then the network would simply perform the identity operation. That is, it would keep any value that you put in indefinitely. So that's the basic idea. The basic idea is to push part of the memory units to this identity configuration. So we push part of the of our autoregressive matrix to diagonal 1 entries and the corresponding W entries to 0 entries, so that these units, by optimization during training, would become to function as memory units. But of course, we do that only for some fraction of all units, because we want to keep the other units free for doing whatever computation we want to, or any task we want to perform. So that's the basic idea, this is just to illustrate that this approach, the regularized piecewise linear recurrent neural network, can indeed outperform many other methods, including LSTMs, on a range of benchmark problems, just illustrating two of them here. A multiplication problem is the same as addition, only with multiplication, obviously. So the bottom line at this point is that we have designed a recurrent neural network with a simple, tractable mathematical structure, yet performance that is at least on par with LSTMs. Okay, so line attractors are not only good for solving the memory problem, but they can also realize arbitrary time constants if I, in my system, if I detune them only slightly. So that's another thing we have published on already 20 years ago or so, and which we will use later on, a property which we will use later on. Also, this is just to illustrate that if we translate now our systems in continuous time, you can derive these continuous flow fields, and you actually can derive a lot of insight on how your recurrent neural network solves the task. So this is for the addition problem, and you can see that it basically integrates the inputs, and then it stores the outcomes on the line attractor dimension. Okay, now let's go. Let's move one step further. So we'll now embed these systems into a generative framework. So the idea is we'll have an underlying latent hidden process, that is my dynamics. We'll have some observations which are produced by my internal dynamics, and we might have some internal stimuli. And we now model this whole system as a generative model that is as a joint probability distribution over these latent states and my observations. And once we have retrieved this this latent dynamics, that's the hope, then we can do all this fancy stuff like, for instance, deriving flow fields and attractive states and so on. Okay, so for the latent dynamical model, I use my generic dynamic systems model that I just introduced, a piecewise linear recurrent neural network. And then the idea is to couple that to some observation model that describes the specific modality we are dealing with. In fact, we'll embed that now into a whole framework. You can couple the same latent recurrent neural network model to a whole variety of different observation models, which capture the different probability distributions of my different observations. So that's briefly illustrated here. So each of these might follow a different probability distribution and might be conditioned in different ways on my latent model. My latent model is my piecewise linear recurrent neural network, now with the Gaussian error term, or equivalently with the conditional Gaussian distribution, where the mean is given by my recurrent neural network formulation. And then the idea is, so we'll have for that Bayesian variation inference algorithms in place, and also expectation maximization algorithms, by which we then, from all the measured modalities that we simultaneously observe, infer the latent states and or the system parameters. And once we have that, we can analyze the recurrent neural networks using the other tools that we have developed. So let me give you one specific example. So we assume that we have some underlying dynamic systems. So for instance, the famous Lorentz weather system, which creates this famous butterfly wing fly structure, the chaotic Lorentz attractor, and irregular time series. So that's my underlying system, but of course, empirically, we don't know the system. We assume we have only access to noisy trajectories or samples, time series samples from that system. So what we do in the first step is we infer our recurrent neural network model from these time series data. Then the idea is, let's forget about the data. Once we have trained our model on these data, let's generate new trajectories from that model. And what we want to have is, and that's here is a real training example, that our model reproduces the actual attractor geometry in state space and the same temporal structure as the original time series. So it's a generative model. Once I have trained it, I can sample from it, and the sample should have the same statistical properties as the original data. And because it's chaotic, it won't precisely overlap, but just have the same temporal structure. Okay, so another highly important point in our context is that often as a metric, if people in machine learning evaluate the systems, they test the prediction error. But prediction errors on time series might not be the best statistics to use if you are in a chaotic regime. So these are just two trajectories drawn from the same Lorentz system with slightly different initial conditions. And of course, because this is a chaotic system, you will have exponential divergence at some point. Yeah, so the time series won't agree anymore. So that's trivial. People in nonlinear dynamics know that for a while, but because of that, we thought of different metrics. We'll take as one important metric in our case the Kullback-Leibler divergence between the true distribution in observation space or projected into the latent space and the generated distribution of our observations given my simulated latent states. Yeah, so this is a generated distribution from the model. So this gives an example where you have relatively high mean squared error because the time series do not overlap, but the attractor geometries agree very well. And this gives another example where our reconstructed system captures the basic frequency of the Lorentz attractor quite well. And for that reason, the mean squared error is actually not that bad, but it does a lousy job in capturing the attractor geometry. So we think that's a very highly important distinction if you want to reconstruct, if you are really after the underlying nonlinear dynamic system. Okay, so this here is just another benchmark which is supposed to show that you can do that now with multimodal data. So it's again the Lorentz attractor, but in this case we made the observations really noisy, or we kicked out dimensions, but instead we assigned class labels to different orphans. So the multimodal system has access to these class labels as well. And this is just to show that basically our approach works and can also use categorical information to infer something about the underlying dynamic system. So this comes back to a point that I made earlier on the slow time scales in the system. So this is an example of a biophysical bursting neuron model where we have very different time scales. We have these slow oscillations that define my bursting behavior, and we have these these very fast spikes. And actually, if you train these piecewise linear recurrent neural networks in this generative model context that I have illustrated, you see that it actually does a fairly good job on this quite challenging problem. And we could also show that this regularization, this line attractor regularization that I introduced earlier, has a significant contribution to that. So if you increase the regularization parameter here, the tau, then you get a lower Kullback-Leibler divergence, and that affects mainly the slow frequencies. So in which you have these slower signals that develop on longer timescales going on. Okay, so now to the last part, applications in psychiatry. In this case, we want to infer a piecewise linear recurrent neural network generative model from fMRI observations. So we have an observation model that takes the hemodynamic response filtering into account. So this is an example from tasks that Florian Biener has run at that time in Andreas Marlinberg's group. It's sort of a human version of this radial arm maze that I have briefly shown earlier. So it's a mutable item working memory task. Again, I don't want to go into the details. People have to remember some stuff, then there's a delay, and then they have to recall it. So this here gives a glimpse of an internal representation of a recurrent neural network trained on fMRI data from human subjects. So what we found not that trivial, at least we didn't get that as easily with other more conventional dimensionality reduction techniques, as that you can see that trajectories in these retrieved state spaces seem to venture into different regions depending on the task period the subjects are currently in. Okay, so this is just some examples of observed and predicted Bolt signals. Also our generative models, and I emphasize again, we do not fit the data. We'll generate new trajectories from the model and then we'll investigate them. We simulate the model after training. And this just shows, for instance, that the model can retrieve major frequency bands of the power spectrum that you have in the fMRI, for instance. We also find that there are a lot of interesting nonlinear dynamical phenomena going on. So, for instance, this is an example from a subject where we found these three coexisting limit cycles of different complexity. Sometimes we find limit cycles and chaotic attractors or other multistability between other dynamical objects. They have various relations to behavior. So this is just one example where we found, for instance, that on the x-axis here, you have behavioral performance. So if the behavioral performance is high, you usually get less stable states and more unstable states, which seems to indicate that higher state space complexity somehow is correlated with better performance on this task. Now, let me give you one more specific example for schizophrenia. So this Florian Biener has run this task also with schizophrenics and with healthy controls. As I told you in the beginning, you can analyze these models partly analytically. So, for instance, you can access all the fixed points and their eigenvalue spectrum. So this here shows histogram of the maximum absolute eigenvalue for controls and schizophrenics. And just as a reminder, if they cluster around 1, then you are around neutral stability. You are sort of in the vicinity of a stable node. But as they go above 1, you have an unstable node or repeller. You can still have limit cycles and other stuff, of course. But what we observed is, interestingly, that you have this significant change in the histogram of eigenvalues. And that seems to be correlated, seems to be rooted in the fact that once we inferred the connectivity matrices, we found in this piecewise linear recurrent neural networks that both the inhibitory and the excitatory weights are usually much higher in schizophrenics than in healthy controls. And that fits very well with the less stability, because that's if all the matrix entries are high in magnitude, you would expect larger oscillations and less stability. And this is just to show that both the negative and the positive weights also correlate with, for instance, the PANS psychopathology scale. So I don't want to overemphasize these data. At this point, it's still preliminary, but it sort of illustrates what you can do with these types of models and this type of approach. So with that, I'm at the end. So my take-homes, I gave you some arguments why I think dynamic system theory is sort of a central layer for connecting neural and behavioral phenomena. I talked quite a bit about our framework of generative recurrent neural networks and how they enable reconstruction of the underlying nonlinear dynamic system. And what I haven't talked that much about is, I just gave a bit of a glimpse, is how you can use it for diagnosis, or you can also use it for, once you have estimated such a model, once you have it in place, you can also use it for simulation of interventions, for instance. And with that, I want to thank all my collaborators, in this case, mainly Heike Tost, Florian Behner, Andreas Meyer-Lindenberg, people from our group. Georgia Copper has done all the fMRI analysis. Dominic Schmidt and Feline Bommer have worked on the machine learning examples that I have illustrated today, and the algorithmic part. Sarah Montferrat is a mathematician, worked more on the mathematical analysis, and our funders, German Science Foundation and BMBF. So thanks for your attention.
Thank you very much, Daniel. So we have a couple of questions for you. Let me go get them. Okay, so the first one, let me see. Okay, so given the dramatic non-stationarity of neural systems, perhaps attractors are not the best metaphor. What alternative ways of thinking might there be in terms of, for example, activity flow, synfire chains, avalanches?
So, attractors are not the metaphor. They are mathematical phenomena that you get under certain conditions. Synfire chains might be explained by these heteroclinic channels. I don't think, necessarily of attractors. It could be transient dynamics. I'm perfectly happy with this. I actually find this idea of heteroclinic channels and orbits quite attractive. So it doesn't, I'm not sold out on that you really need to have stable attractor states. Yeah, and the methodology we developed can retrieve any of these. It really gives you the flow fit. Yeah, so whatever attractors you have in there.
Thanks. So the next question is there a proof of concept that with a small or unexpected, not mainstream change in the dynamics of a psychiatric disease, we can actually change the outcome for a patient?
I think that's too early. Yeah. So at least from our point of view, I think we are right now in the process of applying that to to brain data from different psychiatric conditions or to smartphone data. But I think that's too early to say. Perhaps one more thing on the non-stationarity. The model accounts for non-stationarity in the parameters as well.
So it can model the transitions, you mean, or?
Yes, so I mean, transitions you also get without non-stationarity. If you move between different attractor states, for instance, or if you have like heteroclinic channels, then you have transients, right? Those you can get even without non-stationarity. But non-stationarity is accounted for in some formulations of our models explicitly as variations in parameters.
So the next question is, are there any hypotheses based on these ideas that could demonstrate how medications, for example, ADD, could affect the strength of an attractor state? In other words, any ideas on how this could bridge back to the biology that instantiates these dynamical systems?
that is our idea, right? So that's where we want to get to. We want to, once we have extracted these models, we want to use them to simulate different conditions, and from those predict potentially the most efficient medications. But again, I really should emphasize, took a while to develop this methodology. We are now at the stage only beginning to apply it to different psychiatric conditions.
Okay, I still have a couple of more. So the next one is using the techniques you mentioned, can we integrate different modalities of data, for example, neuroimaging and behavioral data, in order to capture the dynamics of a psychiatric disease more realistically?
Yeah. So that's the framework that I illustrated in the center. So what we have developed is this tool that you can couple the same latent dynamic systems models to multiple modality-specific observation models. And so if you have simultaneously observed data, then you can couple them to the same latent model.
Thanks. So next question is, are recurrent neural networks superior to time delay embeddings for attractor reconstruction? Are time delay embeddings old news now?
So time delay embeddings don't give you the flow field, right? They don't give you a model, they don't give you equations, they don't give you a flow field. They only allow you to sort of retrieve the attracting object for an empirically observed trajectory. Our goal is to have the generating equations so we can analyze them in detail, and to have the whole flow field, and to have basically the whole space, even if they are not completely covered by my experimentally observed trajectory. So that's the main point here.
Thanks. Okay, so I have one more question, and that is, how are the latent variables identified by RNN related to or different from those by other methods like ICA or energy landscape analysis? So
again, ICA won't give you a model and won't give you a flow field. Yeah, so it gives you a model, but it's a linear. So it's nonlinear in the inference, but it's a linear projection matrix. So it will not produce. It's not a time series model. It's not a dynamical model. It won't give you dynamics.
Thanks. So that was the end of the questions, and thanks again very much for the interesting talk. Thanks for joining. Okay, so we'll move to the next speaker, and I just wanted to remind everybody that we have that Google Doc for further discussion. So I will post the link again in case people want to add there. And now I'll invite Gunnar again. And hopefully this time it will be possible to view his screen properly. Let me see. Okay. Okay, I invited Gunnar. Okay, we can see your video in your sound. Can you tell?
Yeah, let's see. Can you hear me?
Yeah. And we can see the screen now.
You can see the screen. This is the most promising so far. So let's see. You see something just white now?
Yeah.
Okay, good. Great, but then I start.
Great, thanks.
Okay. Hi, everybody, and thanks for giving me a third shot at making this work. I hope I can make up for it by having an interesting presentation. And I will talk about a concept which is M4 health in the brain. And M4 health can mean models for health, but it's also a specific type of models that we are doing. So it's mechanistic models, so typically ordinary differential equation, or and then multi-level, multi-timescale, and multi-species models. And we create these models by basically laying the puzzle of, in general, the human body, but here specifically the brain, So we sort how to combine different pieces of knowledge, data and prior knowledge into multi-level models incorporating all of the main organs in the body. And this should be useful for medical professionals, for drug development. We collaborate with AstraZeneca, and for general people that just want to keep track of their health or improve their health. And I will talk about primarily something called the neurovascular coupling, but I will also, in the end, talk about how we connect this to the whole body level, which is digital twins, so computer copies of a person, which is the basis of a spin-off company that we formed as well. And the name of this is Sund, which is the last part of my name, which means healthy. Or that you do something in a sound way. So it's the same word as sound in English. Primarily, I'm employed at Linköping University, which actually is not famous for very much, but we are the most popular university in the world among students. We've been ranked number one across all 5,000 universities that were involved. And much of the things I would present here is actually done by students. And if the logotype for our spin-off company is up here to the right, the logotype for our research group is down here down to the right. And it has two symbols which basically combine the two previous talks. Or is sort of a commentary on the two previous talks. So we do mechanistic modeling, meaning that you can see inside what happens. But we also keep track of which parts of the model that can be well determined, so given the available data. And in fact, we combine our mechanistic models with machine learning models to have hybrid models. So this is a sort of one-slide summary of my presentation. And now let's dive into some more details. Short history of modeling of biological systems in general. Hodgkin-Huxley models, modeling the electrophysiology of electrophysiologically active cells, which has, for instance, been a part now of multi-level models in the heart, which also are connected to blood flow, but also in the brain, neural networks, which can be physiologically based. And in, for instance, Mark Sager's baby X, it's connected to an actual physiological baby that can interact with you, that can learn to recognize you, that cries if you are making it upset and so on. So both of these are very cool, but they aren't really connected with each other. And one of the primary ways of connecting them is the neurovascular coupling. So how does the blood flow that comes to the brain, how is that altered by brain activity? And the neurovascular coupling is also important because it is the basis of fMRI and NIRS data, which typically are analyzed by quantifying each voxel in these type of images to get this type of time series. And then you do a correlation analysis with an archetypical shape of the bold response, which is the signal that you measure. And if you have a high correlation, you get an active area. And if you have a low correlation, you say that there's no activity in this part of the brain, in this voxel. And the problem with this is that this ignores all of the known biology. What we propose here is to instead have a mechanistic model that describes the processes involved. And then by fitting the model to data for the bold response, but also to many other things, you can make predictions. And this can give you a much more nuanced and versatile and reliable activity measurement, and also other things than activity. But to do this, we need to develop a reliable model for the neurovascular coupling. And this is what I will talk about now today. And the way we do this is by taking experimental data and mechanistic hypotheses, formulating them as ordinary differential equations or as differential algebraic equations, And then either we reject the hypothesis because they can't explain the data, or if they can explain the data, we always test the model a couple of times more by doing predictions, and then we save some data or ideally collect new data, which are done as a basis of these predictions. And only if the model can correctly predict new data do we say that it has some merit, and then we incorporate it into the big picture, which we then use for things in the clinic or for drug development and so on. And in the beginning, of course, you need to have a model for the big picture, and for the neurovascular coupling, we'll essentially put this big picture first, and then we'll fill in more details after a while. So coming then to this neurovascular coupling, or the fMRI signal. The first belief of how this works was that it involves a negative feedback. So the reason why the signal goes up is that it first goes down. So first you have a nerve activity, which leads to metabolism, which leads to increased blood flow, which then brings in new glucose and new oxygen and so on. But lately, another hypothesis has replaced this one from the 80s, the original hypothesis, which is a feed-forward hypothesis, which says that this is not acting as a control system in terms of a feedback loop, but as a feed-forward signal, where you have nerve activity which just kickstarts a system, which then just runs its course. And this has been proposed for around 10 years approximately. But neither of these hypotheses had previously been tested with modeling. So this is what we did using our approach. So we formulated first a negative feedback hypothesis, and while it could describe the data, while it could produce this archetypical shape, so here you have actual data, which are the different error bars here, and the simulation with such a model is the line here. So you see it has a good agreement with data, but when you look at predictions, they are widely unrealistic. So for instance, here, glucose is the controlled variable, and then glucose goes down to 0 for several seconds. And this is not what happens, even though we can't measure it with a high accuracy, we know that it doesn't behave like this. And then when we thought a little bit more about it, we realized that, well, this is actually what is to be expected of the control system. So if the control system kicks in because the system initially moves down because you consume glucose and oxygen, then what would happen is that it brings it up, but then once it is up above basal, it would go down again. And here you don't see that. You see this extreme overcompensation, and then it leads to a sort of post-peak undershoot as well. And this is, while maybe the post-peak undershoot would be expected, a complete overcompensation is not how a control system typically works. It's much more of a positive feedback. And in the end, we included the metabolism in the model. So this is the biology simplified, and here is the mathematical model. So each box here represents a state in the differential equations. And we published this in 2016 for the positive bold response, which is the one I showed you. But the year after, we modified the model to also be able to describe something called the negative bold response. And this we did by adding the variable GABA, which leads to inhibition on the electrophysiological level. And in both these papers, we validated the model by experimentally testing predictions of the model, so see what happens in new experiments. So this is agreement of the model with training data for the positive bold response. Again, the simulation is the blue line, and the data are the different error bars. And for the negative bold response, which is here to the right. And then we saw what happens if you, for instance, add a second stimulation. You show the persons a second white circle with a certain time delay. Then the prediction is that it will instead look something like this, which was validated by new experiments that we did afterwards. And the same was true for all the other things that I will show you. So this basically argues that there is a positive feed-forward cascade going on, but that just says sort of on a phenomenological level what happens. If you want to dive into the biology, we need to move from humans to animals, because there you can do much more in terms of unraveling the mechanisms. But the problem with animals is that they are typically, or they are often under anesthetics. And then the question is, what does anesthesia do? So we needed to sort that out first. And there are different hypotheses. One is that it alters the neuronal activity, and another is that it also has effects on the neurovascular intracellular processes. So we identified some data for this and formulated a more physiologically based models for these different arms, these different feed-forward arms. So here we have pyramidal cells and 2 interneurons. And then we have these type of data. And what you also can do in animals, especially in mice and rats, is that you can do something called optogenetics. Which is where you genetically modify the animal to be able to use light laser to excite only a specific cell type and only in a specific part of the brain. So you can be very selective in what you activate, and then by inhibiting crosstalk, you can sort of also not have secondary effects, or you can have it, and you can compare it also with sensory activation. And all of these things give rise to different types of responses. You alter this bold response, which is here approximated by the dilation of the arterioles, so the small arteries. And you see that sometimes you have a longer post-peak undershoot, sometimes you don't have it at all, the amplitudes are different, and so on. So we developed a model for these data. Here you have the 3 cell types again. Here you have the electrical activity, and here you have the control of the diameter of the arterioles. And then we did it in the same way as before. We trained the model to some data. So experimental data, uncertainty are the error bars here, and the best model simulation is the red curve. So we had lots of estimation data for all these different conditions, stimulating some cell types, not others, allowing for crosstalk, not having crosstalk, and so on. And then once we had trained the data and said that this is probably how it works, we tested it by adding validation data. And again, here you see comparison between the model predictions, which is the envelope, and the data, which are the error bars here. And you see that the model can correctly predict new data. So then we had a sort of explanation for this data and for these different conditions. Then we could look into the model and see how can you produce all of these different behaviors. And this prolonged post-peak undershoot, where it stays below baseline for a long time in certain conditions, this could be explained by something as simple as a Michaelis-Menten expression. And the basic idea is that you can translate a large difference in an upstream signal. So here you have a large amplitude difference, where the high amplitude also is high for a longer period of time. You can translate that via a Michaelis-Menten step to something which has the same dynamics difference, but which has removed the amplitude difference. So here you have a much longer response, but the amplitudes are essentially the same. And this was what in the model allowed for this behavior. So on the positive arm, so there were these 2 arms, here's the positive arm, you basically see no differences between the 2 conditions. But for the negative arm, that one that was involved in the post-peak undershoot, that one has this behavior where you have a big amplitude difference, which leads with some time delays to also timing difference, and which then via this Michaelis-Menten step leads to time length difference only. And this is why this one goes down to lower values, but it doesn't go for longer time, but it doesn't go down to lower values. So in this way, we can sort of use simulations to unravel the mechanisms and then test predictions. So it's a way to sort of combine biological experiments with hypothesis testing in a systematic way, where you can keep track of many very complicated processes. So negative arm goes on for longer. And what we have done also is that we have gone beyond this model to one that describes all sorts of other data. So not only this optogenetics data and the ordinary bold response, but all sorts of other data from primates, from humans, from rodents, optogenetics, local field potential, oxyhemoglobin, deoxyhemoglobin, total hemoglobin count, cerebral blood flow, cerebral blood volume, and the BOLD. So we have a single model that can describe data from all of these, from humans, rodents, and primates. And here is an overview of the model. Here we have the pyramidal cells, the two interneurons, and here we have control of the arterials, which then propagates using a blood flow model, which keeps track of the physics, essentially. And then we have the BOLD signal, which is another type of model, which is basically an MRI model. And this is, to our knowledge, the first model that can describe all of these different things. So there are some different previous models that you see here, but they can explain parts of these. Some can explain intracellular mechanisms, but not other things, and so on. But our final model can explain all of these different type of processes, all of these different types of data. And this allows us, again, to explain all of these complicated behaviors by looking into the model. So here we can see if you go from a short stimulus to a long stimulus, you can go from a 1-peak response to a 2-peak response. The first peak is explained by the NO interneurons, one of the interneurons here, and the second peak is explained by the pyramidal cells, and the post-peak undershoot is explained by the NPY interneurons. So we're also doing all sorts of other projects in the neuro field, which I'm not talking about now because of lack of time. So we are connecting our neurovascular coupling models with more detailed electrophysiological models to explain also other data, EEG and MEG and so on. We are looking at social medicine analysis to look at stress and other risk factors. Thought processes according to think aloud protocols, where patients are basically saying what they are thinking, and then we monitor both bodily functions and neuronal activity in different ways. And we are describing then the thought processes combined with the physiology. Stroke, we're doing quite a lot of. Facilitation, which is an intracellular process involved in epilepsy. Metabolism, and also we are moving more and more to interconnected models where we have different brain areas. But let me talk shortly here in the end about how we connect this to the whole body level. So this is our patient 0, the first patient that we did a patient-specific multi-level whole body model for. So we put him in an MRI, and we measured fat content and muscle content, blood sugar meal responses. So here you see comparison between data and simulations. Again, we subdivide the data by estimation. So the lunch is the estimation, and then the dinner response, which is the second, is the validation, where we test the model, and then we do predictions with uncertainty. This model that is actually approved by the FDA as a replacement for animal experiments when certifying certain insulin pumps. Heart and blood flow models, brain activity, stress responses, exercise, and so on. All of this has gone into the same multi-level, multi-organ model, which we are now moving towards testing in the clinic for better pedagogics, personalized diagnosis, better compliance, and prevention. This technology, which is also part of our spin-off company, was launched last year, and that led to keynotes in various places. And if you want to see more of this, here is a YouTube link. But just as here is our first prototype of an interface. So here you can choose. So now the model for this patient 0 is in behind. You can choose which organ to look at. So here we pick the brain, and then you can choose all sorts of different aspects to look at. Now we pick the BOLD signal. You can choose to look at the blood, any blood vessel in the whole body. And here we pick a blood vessel which is close to the, which is located in the brain. And then you basically decide what do you want to simulate for this digital twin of this patient 0. And here we simulate something simple, but which is connected to my first slide. So it's just some heartbeats and some brain activity. And then once you have specified here to the right what you want to do, what you want to simulate, then you can push play. It's sent to a backend where all the simulations are being performed and where the digital twin is stored. And then you get simulations out. Hopefully here. Well, I don't know why it stopped. Maybe it. Yeah, okay. So here is the bold signal, which is very similar to data we have, but we also have blood flow and so on. Somehow something happened with the video. But all of this is on the YouTube link that I showed you, so you can see all sorts of performances. So to sum up here, we have this technology, which we now are publishing and making public so others can use it. And a new version of the software is being released soon, and we will test it in the clinic next year for preventive measures first, and then for various patient groups like type 2 diabetes, stroke, high blood pressure. So to sum up, I presented this concept, M4 Health, mechanistic, multi-level, whole body down to intracellular, multi-timescale, short timescale, milliseconds to years, and multi-species models of the brain and the body. We have modeled the neurovascular couplings in a more comprehensive way than, according to what we know, any other group has done. And this can be used to bridge electrophysiology to fMRI and provide improved merging analysis with a wider variety of different brain measurements. And this is part of a new digital twin technology, where you can combine models and data and knowledge from a wide variety of things. And just some acknowledgement. So the main part of this work has been done by Sebastian that I presented now, but we are collaborating with many different collaborators. So this was my research group. These are the collaborators. Here is some of the funding. Here are some of the key people in the brain modeling. And this is our schedule for how to do the analysis or how to do our collaborations. So that was it for my presentation.
Thanks very much. That was an impressive amount of different systems integrated and scales I've ever seen. So we have a question from the audience. The first question is, what level of anatomical structural details are these models, do these models use, and are they able to use, and what are the rationales for the choices made?
So for the brain, we don't have anything of that at the moment. So we only describe what happens in the voxel. So we sort of assume that this is a well-mixed compartment. But what we want to do now, and what we are setting up collaborations with anybody that we can find, is people who have more whole brain models, and that describes communication with different brain areas. For some of the other organs, we have a much more high resolution anatomical description. So for the liver and for the heart and so on, there are much more widely developed models included.
And you mentioned cross-species models too, modeling across different species. Do you have fMRI for which animals do you have for?
We have fMRI for rodents, humans, and monkeys, so primates. And it's not our data; it's our collaborators' data, or just published publicly available data.
Thanks. So we have another question. What about glial cells, signals from glial cells? Would it be meaningful to include that in the model?
Yeah, we had it. We have had it in some versions of the model. We don't have it in this model because we don't need it, but in principle we could have it. And for the metabolism, it's more important.
Thanks. Okay, so I don't see other questions. Bill, do you have? Oh, you're muted. Having trouble unmuting. Okay, so thank you to all the speakers, and thank you, Gunnar. So we're going to have a lunch break until 2:45 p.m. And after that, the talks will continue at the same URL.
Thank you.
Okay, thanks a lot.